All right, so Bio 30S, welcome back. We ended off the last little part here at the mitochondria. Again, I'm going to keep apologizing, but I didn't realize I have my mouse on my screen the whole time, but we're going to work with this equally annoying laser pointer instead and continue on with the mitochondria. So we finished off the vacuole the other day, and now we're going to look into what you guys might remember as the powerhouse of the cell, or that's what we tend to call it in middle school. This is an organelle where cellular respiration is going to happen. So that's where the name powerhouse of the cell comes from. This is where all the energy um, can be metabolized. So it's typically one of the bigger parts of your cell. Like you're going to be able to see the mitochondria before most of the other organelles that are in there. You're also going to see a lot of these. Depending on what kind of cell you have, you might want more mitochondria if you need more energy in a certain part. Cellular respiration, this is where the chemical energy, so any carbs that you have, it's converted into ATP. And ATP is the energy that we typically use. So the equation, if we were to do some sort of chemical equation for this. And you're not responsible for this in biology, but you'd see this in biochem or in chemistry as well. If you take a carbohydrate, you take a sugar, and you mix it with some oxygen, you mix it with some carbon dioxide, you end up with water and energy. I think I said that wrong. Sorry, let me go back. If you take a carbohydrate and some oxygen, it is going to give you carbon dioxide, water, and some energy. So there should be a little equal sign right here in between the oxygen and the carbon dioxide. So carbs and oxygen, right? We eat foods that have sugars in them. We breathe in oxygen. That is going to be turned into both water and energy, along with the byproduct of carbon dioxide. So we breathe the carbon dioxide out. Our body uses most of the water. Some of the water, if it's a little bit of an excess, we'll flush it through our system. And then the energy is what we use to actually function. Continuing on with the mitochondria. So, cell energy, we can get that from ATP. That stands for adenosine triphosphate. We'll talk more about ATP a little bit later on. Every single organism that carries out aerobic respiration, so they're breathing out in air, except for things like bacteria, will have a mitochondria. At least one, chances are you'll have multiple that are going to make that energy for you. They have a double membrane, so unlike just having the membranes on the other organelles that we have, so like the nuclear envelope, for example, or any membrane around the membranous sacs of the Golgi apparatus. The mitochondria has a double membrane, so it is quite similar to the nucleus in that the inner membrane has a whole bunch of these folds. We call those cristae, and these will project into an inner space. And the space is what we call a matrix. It's just an area. In that area, you've got a whole bunch of enzymes, and that's going to break down the carbohydrate product. So whatever your carbohydrates were, whatever were derived from your carbohydrates, the enzymes, and the enzymes are chemicals that just speed up the rate of a reaction. They're going to help break down those carbohydrates and help us metabolize that product into eventually some sort of energy. In this case, ATP. If you have some cells that need a lot of energy, they're going to have lots of these mitochondria present. So a heart cell, which is a cardiac cell, will have as many as 100 mitochondria in just one cell. So each cell will have one nucleus, but if you have a cardiac cell, it will have almost 100 mitochondria at a time. So ATP production happens in or at the cristae. Um, the way it works is you get these high energy bonds that store the energy, and it's difficult to really wrap your head around this unless you've understood the chemistry behind it as well. But when you have bonds, if you break the bonds, the energy has to come out. So this is what's going to happen in the mitochondria: is you have these high energy bonds that are storing the energy, and these will be broken up in the mitochondria, so the energy is released to the rest of the cell. The cells that have really high energy requirements, so things like heart cells, the cardiac cells, or if you're talking about muscle cells or sperm cells, they're going to have multiple mitochondria because they need lots of energy. As mentioned, the heart cell has about 100 of those mitochondria in there. And that's going to bring us into this other part of the cell called a cytoskeleton. So, as you can imagine, it's some sort of structural support. These are a whole bunch of filaments, a couple of tubules, and they go from the nucleus to the plasma membrane. and it helps give the cell some sort of shape. So it's almost some scaffolding to keep the structure of the cell that allows the organelles to move around, but that prevents the cell itself from collapsing upon itself. Also, what you're going to find on a cell is some sort of cilium or a flagellum. It depends on what cell you have. Some of them won't even have either of these. So we talked about cilia the other day or in the other class. Those are those short little hair things that hang off of the side or the end of some sort of cell body. You can have multiple cilia, you can have just one. It really doesn't matter as long as you have some of them, they're going to aid in the movement of that actual cell. You could also have a flagellum or flagella. These are much longer, you don't get as many of them, but because they're longer, they move in a whip fashion. So think of it like this. If you have a skipping rope, and you spin it over your head in a circle shape, that skipping rope is going to follow that same pattern. It's going to make that circle shape. And if you spin it fast enough, it's almost like a helicopter blade, right? It's spinning around, and that's going to propel you in one direction or another. That's how a flagellum works. It spins around in a circle, it gives you that uniform motion all the way around. It's long enough to do that. A cilia, on the other hand, cannot do that. It's not long enough to spin around in a circle, right? So instead, take the drawstrings from your hoodies, for example. You pulled those off. One, they're too light, and two, they're typically too short to spin around in a circle. So instead, it's easier to just wave those back and forth. Now, as you can tell, that random motion isn't as useful, but if you have multiple cilia on your body, you don't have to move that fast or that far. It's enough to keep your cells moving wherever they need to go. And that's going to bring us into the actual outer part of the cell. So we probably haven't seen this before, but the outer part of the cell is not just a round flat surface like we've been drawing so far. The outer part is going to have a type of membrane. And that membrane, which we mentioned before, is going to either allow things in or prevent things from getting into the cell, and vice versa. It's going to prevent things from leaving the cell. It's going to allow certain things to leave the cell, and that depends what is fixated on that membrane. So a cell, it's not going to survive if it's just itself. Like if it's just a cell, it's not going to do anything. Uh, the membrane itself is going to have a whole bunch of different pieces that are going to help it interact with the outside world or other cells that are around it. So as mentioned, it has a cell membrane, and the cell membrane is going to have little gates all around, and that's going to help us decide and determine what comes out of it. So just in the image that you see over here, in case that's a little confusing, we're zoomed in on the side of a cell, so the edge of the cell, and it would look something like this with the orange dots on the outside and on the inside. Of course, it wouldn't necessarily be colored orange, but it's just for depiction. And then there's going to be these little things that are lodged into the actual membranes. You'll have proteins, things like protein channels. You could have other types of proteins that are there for facilitated movement. You could just have some proteins sitting in the membrane that don't really do much. You could have carbs, you could have some sort of sugar chain, or you could have some sort of fat chains as well, or a combination of the two or others. So lots of different things can get lodged into the cell membrane. Sometimes they are beneficial, sometimes they are not. But we're going to look at a couple that can be useful to us. So the cell membrane is going to decide which substances can pass in or out of the cell. Because a cell can decide that, we say that it is selectively permeable. So permeability is a measure of how well you're able to get things through some sort of membrane or surface or barrier. Selectively permeable means that some things can go through pretty easily, some things cannot. The reason for that is because of the structure of that membrane. It's made up of two different components. One of those are called lipids. Lipids are fats. And the other one, something else we've talked about before, are proteins, a collection of amino acids that chain together to make proteins. The lipids are going to be arranged into a bilayer. And for the most part, if we're talking about plasma membrane, the lipids are phospholipids. So they're phosphorus containing lipids, and they spontaneously arrange themselves into a bilayer. So that's kind of like when you guys come to class. I don't have to tell you anything. You know where your spot is. You just automatically go to your spot. You sit down. Okay, that's how these guys work as well. As soon as a cell has its membrane, it sets up with the phospholipids. They know where they're supposed to go. They just line up. The bilayer itself call it bilayer because there's two parts to it. There's the orange heads, as you can see on the image, but there's also these blue tails on the middle inside part. So the tails are hydrophobic. The word hydrophobic is like a hydrophobia. They're scared of water, and they're going to be pointed to the inside of your cell. And then the orange parts, these are the heads. We're going to say that these are hydrophilic. These are water loving. They're pointed towards the outside. They mix with the outside of the cell and they also mix with the inside of the cell. Now, this makes sense because our cells are floating in some sort of interstitial fluid, which is mostly comprised of water. We drink a lot of water. Also, our cytoplasm, which is on the inside of the cell, is mostly also comprised of water. So we want to make sure that this surface can interact with the water. The surface or the interior surface of the membrane can interact with the water because the polar heads are hydrophilic. The actual layer itself, this inside part, is hydrophobic. So water has a really hard time getting into this space. So yeah, the water can interact with the outside or the inside, but the middle of the layer is protected from the water, so the water can't pass in or out of the cell. It's a really nice system. It allows the cell to be nice with the water, but without letting the water actually into the cell or out of the cell. So that prevents the cell from swelling or shriveling up too quickly. Then we have to work with different ways of getting things in and out. We got these things called glycolipids. They're kind of like phospholipids, except instead of a hydrophilic head, you have a sugar instead. So instead of one of these orange things at the very end, you're going to have what is a sugar head at the very top. And that's going to allow various things to get in. You can also find things like cholesterols. Cholesterols are a type of fat, so they're a lipid. They're also a type of steroid, and they're found mostly in animal plasma or animal plasma membranes. And this is going to reduce the permeability of the membrane, and that makes sense, right? If you replace this hydrophilic head at the top, which is really good with water, and you replace it with something that is like a fat, which is really bad with water, it's less likely that that cell in that region is going to interact with anything in water. And if it's not going to interact with anything in water, it's less likely to have something get in from the water. You've also got what are called glycoproteins. So these are like the glycolipids, but instead of being a sugar connected to a fat, you have a sugar connected to some sort of protein, and these will project outwards, and they have various functions. There's a picture of one in this image here. This green image is, for example, a protein, and there's a chain hanging off the edge of it, which will be a carbohydrate chain, and that might attract certain molecules that are more likely to get into the cell. It might prevent other molecules from getting close to the cell. So it's kind of like a signal to see what should be getting in or out of the cell. So let's take another look at a different image of the membrane. This is if I just look at a side-on view. So you can see that we've got our phospholipid bilayer here, the hydrophilic heads on the outside, the hydrophilic heads on the inside, and then these hydrophobic tails in the middle of the actual cell. Okay, you can see there's the extracellular fluid here, and then you've got the cytoplasm on the inside of the cell. Now, in the images that you see in image A, you're seeing an image of what looks like just a channel to allow these molecules to get from outside to the inside. But in the second option here, should be a B here, but in the second option, you've got what looks like a gate for these molecules to get into before they can actually get in. And that's actually exactly how it works. It depends on what you're actually going to be working with. So in science, we typically work with two temperatures, room temperature or standard temperature, which is 0 degrees Celsius. But when we're talking about the human body, we have to account for the body temperature as well. The body temperature is significantly higher than, say, room temperature or way higher than standard temperature. At body temperature, if you had, let's say, a whole bunch of these cells just laid out on the table and you touch them, it would feel like olive oil. So it's a very soft phospholipid layer. And it makes sense because phospholipid, it's a phosphorus fat layer, and oil is very similar to that. The more unsaturated fatty acids you have, the more fluid that bilayer is going to be because you can have more of those fats in that layer. So if it's more fluid, it's going to flow more easily, and that's going to allow these proteins to float around it more easily as well. And each of the 1/2 layers, so the bilayer is both parts, the top and the bottom combined. The monolayer is just 1/2 of that. What happens is the hydrocarbon tails are going to wiggle, and when they wiggle, the entire phospholipid molecules can move. So this entire cell can move because these tails are always moving around on the inside. They also don't switch their seats. So, in the middle of class, nobody just gets up and swaps seats with somebody else. Once you're seated, once you're in your spot, you tend to stay there, and that's usually what happens. There's a couple of rare instances where, sure, one of the molecules might switch position. That might happen if your cell is under some sort of stress and it has to change shape, but it's so unlikely that that's going to happen. The fluidity allows the cells to be pliable. So because it's fluid, pliability is a measure of how well something can be flexible, how it can change shape. So if the cell has to squish, because it's a fairly fluid layer, it can squish. It's like, if we go back to that balloon analogy, I fill a balloon with water. If you ever seen a water balloon, you can squish and smush it all around because it's very pliable. But instead, if I had an egg, which is also a container holding some sort of fluid, I can't squish and smush an egg because it's got a very non-pliable coating. The shell is very non-pliable, so I can't bend and shape that. Inside of the cell, so inside of the membrane, you can actually hold some of these proteins in place, and it's held by these cytoskeletal filaments. So that is kind of like a scaffolding. It holds the proteins in place. But in the image here, you can see that they're not held together by anything, which means that some of these proteins can just float around in the bilayer and they just move around. That's where they live. They don't stay in one position in particular. But just realize that in some instances, you could have that scaffolding, that cytoskeletal filament that's going to hold it in place. So the membrane proteins, that's going to help us decide what's going to get into or out of the cell. One of those options is what's called a channel protein. That's a doorway. So our classroom has a doorway. That's like a channel protein. Some molecules, if they fit that profile for the channel protein, they're going to be able to come into the classroom. So my door, for example, is a fairly large door. So pretty much anyone can get into this classroom. But I don't know, if you're driving a car, for example, I'm being ridiculous on purpose, but if you're driving a car, you can't get into this classroom because my door is too small for that. Okay, what if I replace my door with a very, very, very narrow door instead? Now people can still get in, but if you're carrying something, it might be harder to actually get into the room. So it's a lot harder to do so. So channel protein is just, it's a hallway. It's almost a bottleneck procedure for only certain molecules to get through. One common channel protein would be an ion channel for a chlorine molecule, for example. So chlorine will form a chloride ion, and chloride is what a lot of our cells will use for that electrical charge. So it'll balance between the neurons when we're trying to send a signal. So it's nice to have a channel protein for those chloride ions. Because we consume a lot of salt as well, chloride is a part of the salt we consume. So it's ideal to have a chloride channel so that we can move that salt from place to place. You can also have what's called a carrier protein, and this is going to interact with a very specific molecule so that it can cross over. So we have a couple of these in our neurons, like the sodium and potassium pumps. Sodium potassium pumps, again, these are involved in that electrical discharge that we have, the depolarization, repolarization. Carrier protein is if I had like an airlock that you had to come into first. So let's say I open my door, you come into the front door, and then I have to check and see if you actually belong in the class. So I'll grab my attendance, I'll look on my attendance and be like, oh yeah, you do belong in the class, come on in. That's what a carrier protein is. You have to fit that certain profile to actually get in. Another example, that would be like a uniform, right? If you're flying a plane and you show up to the airport not in your pilot's gear, no one's going to take you seriously. You're not going to be allowed onto the plane. Even though you're a totally trained pilot, you need to have the right clothes on. Or if you're a police officer and you're trying to get into the back part of a police station or an RCMP office, if you don't have your uniform on, you're not allowed to go back there. So a carrier protein is making sure that you're wearing the right uniform before you get in. We've also got what are called receptor proteins. These are specific shapes, so only a specific molecule can bind to it. So now we're going to use our door analogy again, but instead of having the door open, I'm closing the door. And you need to have a key to get in. So that key holds your receptor protein, and whatever it's going to receive, in this case it's a key, only if you have the key can you get into the room. And so you might have hormones or enzymes that do that. If the hormone or enzyme binds, then it will open up the door, and then you can transfer between the cell and the extracellular fluid. So a cell itself is not going to survive if it's isolated from its environment. It's kind of like you guys. If you're just going to sit at home in your bedroom all day and you never leave, that's going to be kind of tough. Like you're going to go crazy at some point. You have to interact with the outside world. You have to get food. You have to get rid of the waste that's in your room. Um, you have to get some water, and then the social aspect of it as well. So I'm not saying that cells are social in that regard, but a cell needs to interact with its outside environment. It can't just be within its own membrane. And so having that permeability allows it to work with other things that are around it. So cells from all organisms will have that membrane, and that membrane is going to function like a gate. It's going to allow things to get in and out of the cell, whatever the cell needs to interact with its environment. The cell membrane controls how easily these things can pass in and out as well, so it can change that depending on what environment that it's in. So a quick image of things that can leave or enter the cell, just different ways that they can do that. Because it is selectively permeable, these things can also change. So when you're leaving a cell, for example, if you're leaving a cell, you can be secreted out, so you can be oozed out of it. If you have nitrogenous waste, that's something that you need to get out of your cell. There's no reason for having that. You might want to get some oxygen or carbon dioxide out of your cell. You might have too much oxygen. You might have too much carbon dioxide. These are interchangeable. As you can see, you can actually get those to enter your cell as well. And then excess water. If you have extra water in your cell, there's no reason keeping it around as an animal cell. If you're a plant cell, you might store it in your vacuole. But if you have excess water, you'll get that out of your cell as well. If you're trying to get things into your cell, you probably want some nutrients in there to help the aid and development of the different pieces that are in there. You also want your salts there to help send signals back and forth. And then you might want that oxygen or carbon dioxide transfer or that water transfer as well. If you don't have excess water, you're lacking water, or you would like some more fluidity, your cell might allow more water to come in. We actually have hormones in our body that do that as well. Depending on what's in our bodies, that's going to influence how much water gets in or out of the cell. So to stay alive, the cell must exchange these materials on a regular basis. And the cell will do that for you. As long as you're a healthy individual, your cells are going to take care of you. At the same time, you have to make sure you're providing them with all these pieces. For example, if you stop breathing, if you start holding your breath, you're not getting that you're not getting the oxygen into your body anymore. You're not getting that carbon dioxide out of your body. So your cells have a hard time deciding, okay, do I want to let go of the oxygen that's here because I really need it? And they have a hard time releasing that carbon dioxide because they don't know if they're going to get another breath back from that. Or if you're avoiding water or you decide not to drink water, um, obviously you're going to get thirsty. And so you're less likely to have that excess water situation. Your cells are going to send a signal to your brain and make you feel that thirst. Signal goes to your hypothalamus and it tells you that you're thirsty and you get a glass of water.